Today it is my pleasure to present some information to you about navigating adolescence. When we think about adolescence, we're talking about how do we navigate the biology of the brain development, understanding emotions, thinking about mental health, and what might this confer risk for addiction. So if we think about adolescence, one of the things that we know in general, whether you're an adolescent or an adult, we know that in a lot of different capacities, sometimes it really hurts to be human. And so what does that really mean? It means that we think about all the things that we have to navigate through life and the difficulty that it can really place on us. And we know if you think about a lyric by Pink, if you think, this idea of it hurts to be human, a lot of people, as we think about addiction, are really at risk because sometimes people look to substances to help them cope with what can be really hard in life. And so my goal for this presentation is to share with you some of the information we know, some of the neuroscience studies that have been published that have really lended a lot of support and understanding to help us as scientists navigate characterizing brain development, and then in turn taking that information and delivering it back to the public. So if we think about adolescence, we think about a time when people don't necessarily want to talk about their feelings. One thing about doing adolescent brain research is that we can appreciate that for better or for worse, we've all been adolescents at one point. And so, as we go forward and think about adolescence, if you're an adult looking back, you think, oh, what a great time in life that was. It's like skydiving, it's exhilarating. But we also know that that's not the whole story. We know that life can really be a roller coaster ride. You're up one minute, you're down the next. It is a real emotional roller coaster. But then, of course, I would be remiss if I left out the influence of peers. Nobody wants to be the odd duck out. Do I have the right cell phone? Do I have the right genes? And maybe for scientists, we think about genes in a different way. But the idea is that this time in life, this exhilarating time in life, that's also faced with a lot of emotional storminess and a lot of peer influence, we know that this is a really important time for gaining new experiences and ultimately making good decisions, because the goal of adolescence is to reach adulthood, how to become independent. And so in my laboratory and a number of other laboratories, we're really dedicated to understanding the biology of the brain and what factors about the brain we can begin to put together to understand, number one, this really dramatic part of the brain development that occurs really primarily in the second decade of life. And if we think about it, there are aspects of adolescence, brain development, and risk-taking and novelty-seeking and decision-making or working on decision-making that actually behaviorally has some overlaps with things like addiction, some aspects of addiction and some aspects of some mental health conditions. Some of those examples are things like risk-taking and impulsivity. By design, an adolescent is risky and impulsive in a way to get them out of the nest, to seek new experiences so they can learn new information and ultimately become independent. On the other side, if we think about addiction or if we think about some mental health conditions, we also know that there's risk-taking and impulsivity and sometimes some suboptimal decision-making. So how do we take all three of these elements together and appreciate the biology of the brain? And so the goal for the presentation today is to think about the structure of the brain, the function of the brain, other aspects of brain, but also aspects of emotion regulation and decision-making that really kind of confer. So we think about adolescence as this time of dramatic brain changes. And this is not necessarily new information, but what makes this new is the technology to be able to understand it and to study it. MRI has been around for, a number of decades at this point. It's become more available, but is one of the best tools that we've been able to use because MRI is entirely non-invasive. So if we want to set out to understand the human adolescent brain, we certainly want to use tools that don't cause harm themselves. And so over the last 2 decades, there has been a dramatic increase in how many people are using this technology to be able to have a window into the adolescent brain. Some of the things we know that, again, might not sound necessarily surprising is that from the time you're born until you reach age around 5 or 6, the size of your brain plateaus significantly. Oh, but by the time you reach age 6, your brain actually doesn't get any bigger for the rest of your life. And so you think about a 6-year-old versus maybe a 12-year-old, and you think, of course they're smarter. They can do more things. Their brains must be bigger. But it's not about the size of the brain. It's about the connections of the brain. It's about the sculpting of the brain. And so if by the time you're 6, your brain has gotten as big as it can be for the rest of your life, how does the brain get smarter? How do we make decisions? How do we have new experiences? Part of that really has to do with this fantastic symphony, this reconstruction process that occurs over the second decade of life. And so, the other thing that we know primarily from imaging studies over the last couple decades is really confirming what we've known for a long time about the brain, but ways that we couldn't study in real time or in real life. But now we know that not only does the brain change significantly over the second decade of life, it also doesn't finish. We used to conventionally think that we reached adulthood by the time that we were 18, at least in the US as a legal age. But one of the things we know about the brain, and we'll talk about this more and what that actually looks like, is that even from 18 to 25 and in some conditions beyond, the brain continues to change and grow. And that is the hope, right? We have this fantastic brain that is resilient and redundant for many reasons. And so when we talk about this reorganization project, what are we really talking about? This is the neurobiology 101 of how the brain develops. So we are born with billions of neurons, more than you will ever have in your lifetime. And so how does the brain change? There are two main processes that really are occurring. So if we take those billions of neurons, what we know is that two things have to happen. And this is really accentuated through early development as well as during adolescence. One of the things we know is that we have something called cell pruning. So we're born with all of these brains, and slowly over time, through genetics and through environmental, interactions, we actually prune away the neurons that we don't need. So our brains are built with redundancy. And so essentially, we want to keep the best of the best neurons, and we want to prune away the ones that we don't need. That is the most efficient way to run a brain. But that's only one side of the story. The other side of the story is that there is also something called myelination. And so what myelination is, it is taking neuron A and forming a connection with neuron B. It's taking the connections between them, the axons, and actually wrapping them with an insulation. By insulating these axons, connections between neurons, we're actually allowing the message to go from neuron A to neuron B significantly faster and lose less information along the way. And so this is how we build better connection in the brain. So we're keeping the best of the best neurons, and we're making them really good communicators. Some neuroscientists might like to say this is how you get better cell service. So in an effort to understand what this looks like, even if you are not a skilled neuroscientist or look at brain images, in this image, as if someone were looking straight ahead at you, you can appreciate that there are different parts of the brain. There are the cells themselves, or the neurons. That is also called gray matter, appears gray in MRI images. That is the band around the outside of the brain. And then there are the connections between the neurons. This is called white matter. This represents myelination, or that insulation around the connections. And one of the things that we know that, again, pretty dramatically, even for people who are not neuroscientists, you can appreciate the difference between the developing brain, when you have a lot of neurons and a little bit of connection between them, and slowly over time, largely the second decade of life, we know that now we have less neurons, we've pruned away the ones we don't need, and we've made better connections. So just to show that to you one more time, you can see you have a lot of neurons and not a lot of connections, and slowly, largely over the second decade of life, we've pruned away the ones we don't need, and at the same time, we've made the best of the best better communicators. So this is one of the neurobiological processes when we talk about brain biology and what happens over the course of the first 2 decades of life that ultimately help people become smarter, make good decisions, ultimately become independent, is that even though we start as infants, we know that we're highly dependent on our caretakers over this span of time. And again, this is work that a lot of researchers across the world have been doing to really try to understand this process. So ultimately, how do we understand going from being a dependent child or infant to someone who looks like an adult, who has grown tall, who has gotten, to a point in their life where they're making some decisions, with the ultimate question being, okay, so when do we reach neurobiological adulthood? And what we know from these MRI studies is that very convincingly, and with a lot of rigor and reproducibility, we know that the brain starts to conclude or plateau this pruning and myelinating into the mid-20s. And everyone is an individual. 20 is not 20 for everyone. People who are biologically female versus biologically male, there are different courses of brain development over this course of time. But the idea being that we know that in the first decade of life, Infants and children are dependent on their caretakers. In the second decade of life, there are more skills, there are more capabilities, but how do we then go to the next stage in life where people can apply those skills to be able to make good decisions and live healthy lives? And so if we think about all of the amazing things that the brain does, so some people have said, is it true that you only ever use 10% of your brain? That is a myth. We use all of our brain all the time. It is truly a fantastic symphony of connections between different parts of the brain. And to the level of advance in technologies these days, we're just beginning to understand the complexity about how the brain does talk to itself. But when we talk about the brain development during the adolescent period, while the brain is pruning and myelinating across all of the brain, there is one part of the brain that really stands out in terms of doing the most rapid developing, and that is the frontal lobe. And if you think about the frontal lobe, the area of the brain that's right behind your forehead, we think about all the skills, the functions that are carried out by our frontal lobe. These are called the executive functions, and they include things like paying attention, working memory, abstract thought, cognitive control, judgment, reasoning, and ultimately decision-making. So the pruning and myelinating, while it occurs across most of the brain, it really reaches its plateau. It's the last part of the brain to develop. So as we think about it, the last part of the brain that is coming online is also the part of the brain that is going to provide us with the executive functioning skills that are necessary to reach that next level of independence. And so some of the things that we're very interested in, and a lot of others, is how does the process, we talked about the structure of the brain, the neurons themselves, the connections between them. Imaging also, MRI also provides you with an ability to look at function of the brain. And one of the ways we do that is by looking at something called cognitive control. So this is an example of a task that we've used in our laboratory. Essentially, you're laying in the MRI scanner, and while we're collecting a signal from your brain, your instructions would be to press a button every time one of the shapes are shown to you. So press a button when you see a large circle, a large square, a small circle. Oh, but when you see that small square, don't press the button. This is the computerized version of Simon Says Do This. And so what we're essentially looking at in the brain is the frontal lobe's ability. Now granted, we do use all of our brain and all of our processes, but it's really required that the frontal lobe be functioning efficiently to be able to perform this task. And one of the things we know is it actually takes more energy of your brain to be able to not respond to something than to actually make a response. So think about all the energy it takes to hold something back as opposed to actually making a response to something. And so one of the things that we've learned is that the frontal lobe is the major brain region that's used during this task, and this efficiency improves with age. If you look at brain images, what this image shows you is that bigger area of activation in the brain is how much more an adult will activate their frontal lobe when performing this type of task where they have to hold back a wrong response, which is don't put your hand up when that small square appears. And so what does that mean? More activation in the brain is not always better. In this particular task, what we know is that more activation of the frontal lobe in adults is actually associated with them being more accurate at the task. But you know what? Teens are really great at this as well. They're very, very fast. And this is kind of the biology of the speed versus accuracy trade-off. So we've learned something about neurons, connections, and redundancy and pruning. We've talked about the frontal lobe activating when we're trying to hold back incorrect responses. This is really the neurobiological basis of cognitive control in decision-making. We know that this gets better with age, and this is the speed versus accuracy trade-off. Over even one year in adolescence, you can see the contributions from the frontal lobe changing to help the adolescent become more accurate. And sometimes the trade-off is time. Another example of this task is something called the Stroop interference task. This is another type of frontal lobe task that essentially asks you to name blocks of color. Not very hard. We learn to name colors early in life. Read words written in black ink, red, green, blue. Not very difficult. But then the last part of the experiment is to hold back the want and need to read, but instead name the color of the ink that the words are written in. Doing this experiment in large audiences is always a lot of fun because it's frustrating and it's really a challenging task. But again, this is the frontal lobe coming online to say, you know what? Naming colors is not very hard. I want you to hold that back, and instead I want you to read the letters. Also not hard, but when you've pitted the two against each other, and that sometimes is what neuroscientists do, we cognitively conflict the brain in an effort to understand the hard work that's going on and the circuitry that's involved. And similar to what we saw in the other computerized Simon says do this task, we see something different. We see that the frontal lobe comes online, lights up, while performing this task. And you might wonder, well, is that all it takes the brain to do to be able to perform this task where you have to hold back the want and need to read? So you might want to say red, but blue, you have to literally hold it back. This conflicts the frontal lobe. And what we know is that the areas of the brain that are lit up in this image are really reflecting the specific type of brain area that you need to effectively hold back. So you're still using your visual system, you're still using other systems in the brain. But we also know that the function of the frontal lobe is linked with how well people do on this task. So at this point, we've really thought about this from a neuroscience perspective. What tasks can we give to people to have a window into the brain to understand how it's working? But what happens in real scenarios? What happens when teens are faced with tough choices? And while we're talking about teens right now, we also want to remember that every adult was a teen at some point in their life. So if we can appreciate the developmental process and understand the vulnerabilities for what might be at stake in the brain, thinking about a tough choice as, okay, what do I do after school today? The bell rings. Oh, you have a math test tomorrow. Well, there are a lot of choices that one could make. A quick response, bell rings, quick response might be, I'm going to hang out with my friends. No thought needed. But if you think it out for a second, perhaps I should study for the test. Because every single response we make is associated with a potentially different outcome. So chances are, if you don't think about it, you blow off studying and you just, hang out with your friends, chances are more likely that you might fail the test. Not always, but just for the sake of this presentation, if you think it out for a minute, go study, and then hang out with friends later, chances are you'll do better on the task. So we spent some time talking about the frontal lobe and how important that part of the brain is for cognitive control. It's not all about the frontal lobe because we have a lot of other parts of the brain that are really, really important. And so in this context, what is this quick response? And I'm sure you can appreciate, you've either said to someone or said to yourself that idea of what were you thinking? So our lab really is interested in the neurobiology of what were you thinking. And it's not just adults saying that to teens. It's adults saying it to themselves. It's teens saying it to themselves. But what we're really talking about in terms of circuitry here is not so much about the frontal lobe. It's about another part of the brain, the amygdala. This is a very primitive part of brain, meaning that it comes online very early in development. And the purpose of the amygdala, it's a structure of the brain that is deep-seated in the middle of the brain, and it's really important for threat detection and survival. So it makes sense that we wouldn't want to think things out. I'm walking down the street and I see a big lion running my way. Does it make sense that I would turn around and be like, oh, that's a lovely lion, I wonder? No. Our system is designed to not have to think, run, survive. And so a lot of research has not only been interested in the frontal lobe and how it confers really good decision-making ability through cognitive control, but also how does the frontal lobe work with the amygdala and other structures to help in the context of things like emotions. Now, I show you pictures of food, not just cheeseburgers or French fries. If you look at pictures of other types of foods, such as oranges or salads, one of the things we know is that if you're laying in a scanner and you look at pictures of food, the amygdala will activate. You don't have to perform a task. Just looking at food stimuli activates that part of the brain because food is related to our survival. But what's really dramatic is if you have a window into the teenage brain, you can see what those splotches represent is activation of the amygdala. That makes sense. This is the part of the brain that is primitively programmed to respond to stimuli that will help promote our survival. Again, maybe not cheeseburgers to some, but the idea is if you have a window into the adult brain shown the same exact images, what you see is some activation of the amygdala down at the bottom side of the brain. We also see something in the adult brain that we don't see activate in the teenage brain, and that is the frontal lobe. So one important takeaway here is that, how an adult perceives a situation perceptually looks very different than how an adolescent might perceive a situation. And so this is not better or worse, this is different. And so that is why, in some ways, we have to serve as a frontal lobe for our adolescents, help them see, help them through learning. And as this frontal lobe is slowly coming online, engage in things like Socratic dialogue. In other words, engage abstract thinking. You're going to go to a party. What would happen if? Have you ever thought about? Help teenagers put these scenarios through their minds and think it out, because the frontal lobe is coming online. There's abstract thought, but then there's also not thinking. There's the quick responding. Maybe they go to a party and maybe someone offers a beer and they don't want to take it. But that frontal lobe is not yet mature and putting the brakes on. The quick response is, I want to be liked by my peers. How do we insert some time into the process? So a good friend and colleague, Dr. Barbara Green on the South Shore, had shared this with me a long time ago, and we've shared this message with parents and with teens, is that you can use something called the 10-10-10 rule. In the 10-10-10 rule, it is a term borrowed from Susie Orman, essentially asserts that wait 10 minutes, 10 hours, 10 days. If you have to make a significant decision, take time. If we think about the concept of time in neuroscience, 10 seconds is all it takes. If I stop talking for 10 seconds, it's going to feel like a very long amount of time. But if a teen goes to a party and is offered something they don't want to do, if they can use something like the 10-10-10 rule, enough time. Hold on a second, I just have to go text my mom. Build in 10 seconds. 10 seconds could be the decision between a better decision and a worse decision. And again, not everyone's brain is the same. So maybe some people need a 12-12-12 rule. And the idea is that what we're trying to overcome is the brain's emotional center. So we talked about something like food, but we also know that the amygdala is activating. We know that the frontal lobe is activating in adults, but what about emotional stimuli? So this is a picture of fearful affect. So what happens when someone sees fear on someone else's face? Again, this is not a task. If you're just passively viewing this, so just laying there looking at it, again, the amygdala will activate, and that happens very early in life. Remember I said that this is a very primitive structure, and the amygdala develops early to help with our survival. So we have this threat detection machine that only slowly over time, the frontal lobe, as it matures, starts to engage to help put the brakes on. So if you see a fearful face, your amygdala will think, threat, 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 get out. Oh, here comes that lion, run. But what if the fire alarm goes off? So the fire alarm goes off and we think about it for a second. That's a threat. We evacuate the building. We wait outside. And then we come back inside and the fire alarm goes off again. Is our amygdala going to respond the same way? It will probably have a reaction, but now the frontal lobe can step in with a little bit of logic and a little bit of experience to know, oh, maybe there's something not working correctly with the fire alarm. The idea is that one of the notable things is not only the frontal lobe developing, but it's also the frontal lobe's ability to put the brakes onto this emotional center of the brain. And if we think about it, for adults and for kids, sad, mad, glad, worried, it's actually not that simple at all. You can be waiting for a text message and the dots are going, and then someone sends you a sad emoji. In a moment's time, not even, your amygdala is activating or some other part of your brain is reacting to the stimulus that you're looking at. And a lot of people have been, looking not only at tasks to see what parts of the brain were engaging, but people have also been trying to map when you're just at rest, if you're laying in a brain scanner, and I'm showing you these images of different type of affect. So if you're looking at sadness or anger or, happiness or worry, where does emotion live in the brain? There's a lot of overlap. A lot of it is in the amygdala. A lot of it is in the frontal lobe, but each emotion has a little bit of a different neurobiological signature. And I think that's really important because emotions can be very confusing sometimes. Under some circumstances, like something being bittersweet, how could something both be happy and sad? And so the idea here is that we're really, as neuroscientists, trying to work towards mapping where emotions also live in the brain. And so, we can think about sad, mad, glad, and worried. It's definitely not that simple. If you look, across all of these emotions, it just expands out more and more. If you look at any of the work of Brené Brown, she has done an exceptional job being able to chart. She's created an atlas of emotions, 87 human emotions and experiences, that the more we learn about what emotions are, if we can actually ascribe an emotion to a feeling, that's actually a really important way to help that frontal lobe connect to the amygdala. And so, one thing to note too is that we're also carrying a lot of very complicated and negative emotions a lot of the times. In her book alone, most of the chapters are about emotions that are hard to deal with. The ones that are highlighted in red squares are the positive emotions. And if anybody here is a fan of Inside Out, highly recommended from this neuroscientist. Great way to think about emotions in the brain. So, as you may know, Inside Out 2 just was released. In the first one, it was really revealed that living inside this 11-year-old girl's brain were these emotions: joy, fear, disgust, sadness, and anger. And while it seems simple, one of the things that became very clear is that joy was in charge of her control panel. And so if you think about it, joy was one single positive emotion. If you think about kids who are starting to navigate and understand and experience new emotions, and adults as well, because this is built when we're young and it carries through our entire life, one of the things we know is that joy has a really important job. And so it takes one positive emotion's work to hold up these harder emotions. One of the things that wasn't surprising that Inside Out 2 actually added in was anxiety. These days, anxiety, we've talked about anxiety forever, but now anxiety really deserves to be its own character. And not surprisingly, as teens are making their way through this developmental period, there are more and more emotions. And one of the interesting things about this second version of this movie is that joy is really squished by a lot more hard emotions. They're all squished in there together. I particularly was a fan of ennui, the boredom, and understanding, that has a role for kids too. Even adults, we don't have many chances to be bored, but there is some value in letting our brains reset for a moment. And so, how do we manage these emotions? If we work with the brain, if we work with the neuroscience, we know taking time can help the frontal lobe process emotions. Even 10 seconds can help. Naming the feeling. If you can name, I'm feeling really sad right now. There have been studies that have shown that people who label their affect, label their feelings or emotions, actually can process them better. And one of the interesting things that came out of the Inside Out movies too is that emotions like sadness are actually very functional and important. So instead of closing it out, letting sadness, letting all the other emotions in, kind of work with our neurobiology is really one of the best practices to think about how we can navigate really tricky times. Because emotions touch everything that we do in every way, shape, and form. And a lot of people talk about mindfulness. Taking 10 seconds is a practice of mindfulness. It's not a 10-hour session, it's 10 seconds. Maybe build to 12 seconds or 20 seconds. But it's a good way to start and kind of reset those connections between the frontal lobe and the amygdala. And so we take all of these pieces, and this is a very relatively long buildup to saying, well, then why is there increased risk for substance use in kids? Well, we already know it's a time of increased vulnerability to substance use and addiction due to biology, psychology, social factors. Right? So there's brain development. That circuitry that's going to help you make good decisions about substance use is only coming online. So if you start drinking or start using drugs before that part of the brain is good at helping you inhibit, that can really have an impact. And it not only impacts the ability, but it also impacts the development. Another big piece, as I alluded to in one of the beginning slides, was peer influence. Not just teens, adults as well are very highly influenced by their peers' attitudes and their behaviors. But this is also a time for impulsivity and risk-taking. And again, this is very important because we think about what one of the evolutionary goals of adolescence is, and that is to leave the nest, right? To become independent. And so part of the brain is wired for risk-taking, novelty-seeking, which might look like impulsivity. Part of that is because of the frontal lobe, but this is part of the neurobiology of what makes the adolescent brain more vulnerable to something like addiction later in life. And also psychological distress. We know that what happens, particularly early on in one's life, so if they have adverse childhood experiences, we know that that can have an impact on brain development itself. And part of that impact is on the frontal lobe and its maturation. Again, it's not necessarily that it doesn't develop, it develops differently. So for someone who's experienced childhood adversity or someone who has a genetic risk for addiction, they might need a 15-second, 15-15-15 rule to be able to help put the frontal lobe brakes on the amygdala. And so psychological distress, coping with stress, coping with anxiety, and other mental health issues, sadness, depression. All of these are things that increase risk for substance use. And also genetics and environment. And while genetics are not destiny necessarily, we know that there is a family history of addiction in the family. It increases the chances of an addiction in the developing offspring. And so some of these things are very important to know. My frontal lobe, how it works with my amygdala. If there is a risk for family history of addiction in my family, then we might need something like a 15-15-15 rule. Maybe not forever. It is a changing dynamic process. But the longer we can protect that second decade of life, that developmental process of pruning and myelinating, the more we have a reduction in risk of an addiction later in life. And then, of course, environmental factors, parental substance use, what kids are exposed to, if there are some sort of family dysfunctions, socioeconomic status, exposure to trauma and adversity. Again, these environmental factors can have a big impact on brain development itself. And if it's impacting brain development, that can impact the ability of the frontal lobe to mature in a way that can be protective against addiction later in life. So, early initiation we know is one of the best risk factors or one of the best predictors of who goes on to have an addiction later in life. And what this looks like is, at least with the case of alcohol, we know that kids start to experiment with alcohol. About 4% start to use alcohol when they're 12, and this increases dramatically to people who currently use alcohol to be about 70% by the time they are 21 years old. And so what does that really mean? We also know that there's a dramatic increase in binge drinking rates, but the age of onset of first use, so for example, from a very large national epidemiologic study, what we know is that the younger you are when you start using, the greater the likelihood of an addiction later. So if you start drinking at age 13, we know that there's about almost a 50% chance of an addiction. If you wait only a few years until 16, this goes down to 32%. If you wait until 18, it goes down to 17%. If you wait until 21, the legal age in the United States to drink, it still is 9%. Having a risk of an addiction to alcohol is never zero unless you are lifetime abstinent. But the cross-section of people who drink in the United States is roughly about a third of people either don't drink or drink in very low amounts. About a third of the country drink in, social levels, and about a third of the country drink at heavy rates of alcohol use. So the idea here is that the longer you can delay the onset of first use, the more protective we can be of that brain biology. So how does this look when we put people in the scanner and we start to understand, what is the brain doing? One of the things we know is that in youth who use alcohol, they perform about 10% worse on tests of learning and memory. So they don't need to be intoxicated for their brains to be different, but exposure to a developing brain during the period of adolescence actually takes away from this developmental process. In this particular example, you would put someone in the scanner, and in this particular work, these are investigators from the University of San Diego, they basically said, when you see that the icon is in the same location, I want you to press a button. And so what they basically were looking at was what part of the brain is activating when this is a working memory task. When you see that they're in the same place, your brain activates. Well, this group, when they looked at people who were drinkers, so they compared teen non-drinkers to teen drinkers, the teen drinkers were not intoxicated when they performed this task, but what you actually can see is that there's a lot less brain activation when they're trying to perform this memory task. And that's really important because we think even for kids, if they drink on a Friday, their learning and memory is going to have a longer residual impact on how their brain is working. The brain is less efficient when it not only has to manage brain development, but it also has to manage any sort of a challenge like alcohol to the system. In a similar way, in the task that you saw, the Stroop task, the cognitive control task, one of the things we know is that if you consider the chicken or the egg, well, teens who are already drinking, I can understand that their brains might be different. But what about teens before they even ever started? And this kind of hints at the idea that if there's a family history of risk, so if you look in teens before they even started drinking, and you compare teens who have a family history of alcohol addiction to those who do not have a family history of alcohol addiction, one of the things you could see is that they don't actually perform the task differently. They scored equally well, but the kids who have the family history of risk, who have an addiction in their family, actually had to activate more frontal lobe to perform at the same level. Again, is this better or worse? No, this is different. It just means that there is something that is genetic that contributes to how the brain is activated. So we're asking the frontal lobe to perform this executive function task. The brains of kids who have a risk for addiction actually have to work slightly more. So it's a different inefficiency. And so that is one of the things that might be related to risk for addiction. If we also consider marijuana use, we know that this has increased dramatically, especially as legalization has changed across the country. We've seen dramatic increases not only in use, but we also know that teens are reporting that drinking is more risky than marijuana use. And so, in terms of harms, they think that using marijuana is less harmful. But there's also another really startling statistic is the relationship between teens who are using marijuana and levels of depression. And so one can ask the question, are kids using marijuana to deal with their depression symptoms, or is marijuana actually causing depression? These are difficult questions to answer, but that is some of where the field is. But one of the things that we do know is that one of the best strategies that we can use is to really share the message and share the neuroscientific evidence that the longer teens can wait to use substances, we're talking about substances that are legal, like alcohol, marijuana, when the brain has reached a developmentally mature age, that is the best strategy for helping to reduce risk for addiction later in life. And so if we look at the same task that we saw in teens who consistently used alcohol, we saw that they had a decrement of about 10% memory issues. In the same group, they also looked at teens who were using marijuana. And when they were trying to perform the task, what was actually found was that teens who had reported using marijuana actually activated less brain when they were supposed to be performing the task. But during the task, when they were supposed to be resting and not performing a task, one of the things that was found was that adolescents who were marijuana users actually now activated more brain. So there's a bit of a mismatch in the efficiency of the brain. So in other words, they activate less brain when they're supposed to be performing the memory task. And when the brain is supposed to be resting and recovering, there is now this mismatch in how much brain is being activated. And so again, with this idea of this cognitive control task, if you look in marijuana users, again, not intoxicated when they perform this task, one of the things we know is that in youth who use marijuana, you can see that there is a very defined, efficient amount of frontal lobe that's being used to perform the task. Significantly more of the brain is activating. So in other words, just exposure to marijuana or to alcohol actually requires the brain to recruit more resources to perform at the same level. And so, one way to think about it is imagine you have two people getting ready to run a 50-meter dash. Only one person has on a backpack of bricks. We can think about that backpack of bricks like substance use in teens. It puts an extra load on the brain that's trying to develop and trying to really be efficient. And so some of the things, again, these are data that have been collected to show that there are not only negative impacts on functions in teens, but it also impacts the development. So it is a cycle on all of the stages. And of course, there's a lot of literature, emerging literature about nicotine and vaping. We know that, there's been a dramatic increase in how many teens are using nicotine, vaping, also vaping, cannabis. This research is really just coming out. But we know with this increasing prevalence, one of the most striking pieces of evidence that's coming out is a really big and not surprising impact on sleep and mental health. So nicotine as a stimulant, we know impacts when the brain can actually recover and rest. And this impact on sleep is really, this connection, again, between the frontal lobe, the amygdala, but also the emotion centers of the brain. Those are really what are tying together not only risk for addiction, because nicotine is a highly addictive compound. We also know that there's also an increased risk for depression, and part of that is thought to come by way of this disruption in sleep. And so, foster good sleep hygiene. I think everybody could stand to sleep better or sleep more. That may actually provide some benefit to teens, not only with sleep problems, but also fending off any sort of addictions, promoting better mental health. And so, of course, we also know that there is an opioid story as well. And one of the things that we have seen is that there is actually a greater prevalence in use of opioid drugs, non-prescription opioid drugs in teen girls relative to teen boys. And so we see this pattern in alcohol also. So girls have risks in some domains, boys have risks in other domains. We also want to appreciate gender identity and the role that, how one views themselves relative to their sex assigned at birth, the impact that has. But again, at the end of the day, these developing brains, regardless of sex or gender identity or background, we know that there is inherent risk just in the biological process. And so really, again, to think about this frontal lobe that is orchestrating all of these really important behaviors. How do we process reward? How do we process social rewards? The thumbs up, the likes. How do we regulate emotions? How do we think about making social comparisons? I didn't get as many likes as this person got. What picture am I putting up? How does that impact my self-esteem? Because all these neurobiological systems, again, really get to this idea of sense of self. And so, the use of substances are particularly important because it impacts the development of healthy regulation of emotions and sense of self and identity. And so cognitive control, attention, a lot of people want to know the question, what impact is social media having? It's hitting each of these different components of what the frontal lobe is working so hard to orchestrate. And, what is rewarding for one is not always rewarding for everyone. If we think about teens who have some level of inherent sadness, depression really appears to alter how these brain connections form. So, you might have some pruning of neurons and some myelinating to make them good connections, but we know that depression, again, we can ask the question, which came first? But we do know that there is a link, that depression, untreated, can actually impact how the brain makes these connections. And so these are really important things to tie together. And, why the increased risk for mental health issues? Mental health and addiction are intrinsically connected to each other. While many adolescents will navigate this stage of development successfully, most adolescents will navigate adolescence without developing an addiction. Who is at greatest risk for the addiction, for a mental health issue? Whereas others may experience challenges that increase their risk of developing mental health disorders. Part of this really has to do with this neurobiological process and what the exposure is in terms of the environment and the contribution of genetics. So again, biological. Is there a genetic family history of mental health issues? That can have an impact on whether or not symptoms of depression or anxiety start to manifest. Psychological, stress and trauma have huge impacts on the development of mental health issues later in life. Of course, social, it's really hard to take any of these apart. Also environmentally, socioeconomic, academic pressures, the media, technology, devices, exposure to cyberbullying, negative online content. We have to really think about a very comprehensive and complex system and figure out the best ways to be able to protect the brain. And so substance use in particular, we know what is kind of occurring when the brain is impacted by substance use. But again, how does experimental and regular use really link to depression, anxiety, psychosis? So people who are drinking or using drugs to cope versus, an expectation that it's going to make them feel better. Maybe they're feeling socially anxious. And so it's reinforced that if I'm socially anxious and I drink, then I feel better. I feel like I fit in better. And so I think we have to appreciate how the brain is developing and what types of, protections we can put in place. And so how do we make sense of all of this? There is so much information coming out all the time. There is a landmark study of 10,000 kids being studied over the course of 10 years, which includes brain scans and clinical assessments and, hormone measurements, media use. And so we're starting to collect data, 10,000 kids measured from age 9 to 10, all the way up for 10 years. And so this is going to provide information to us that will help us see, well, what does the brain look like before someone even starts to feel depressed or anxious? What does the brain look like before someone starts to use substances? What's the contribution of childhood maltreatment or, adversity exposure during childhood? An important thing to consider that I think is really important too is that we need to also ask teens how they're feeling. We need to also ask adults how they're feeling. So in a New York Times article, why are American teens so unhappy? There was actually a response by a teenager who said, people have a tendency to think that social media or my iPhone, quoting the person who wrote the article, they have a tendency to think that it's my iPhone that's causing my depression. But the world is very complicated. We're coming through the end, hopefully, of a pandemic. It is a time for a lot of anxiety, and there's a lot of depression also all around us. So one of the things I liked about this is I think that the voice and the information that we can learn from teens about how they feel and what they're experiencing, because we know when a teen versus an adult looks at a cheeseburger, their brains light up very differently. We also want to know when you see a situation, and just because how it was when you were a teen doesn't necessarily mean that's what teens are experiencing today. And there's a lot of focus and a lot of concern about social media and device use. And, unfortunately, that is our next evolution. It's just around 100 years where we've had electric light. Our brains had to adapt from only being able to be active in the daylight hours because there was no electric light at night. Our brains had to respond and adapt to that. We also didn't have vehicles, so people walked or they rode horses. Now, again, our brains had to respond to the changes of being able to, not only go from here to there in a car, but here to there in a plane. And so this digital evolution, this digital technology age that we're in, one of the things that we can think about is that those brains that are developing, those same brains came up with the new technology. So how can we use this technology to help protect our brains even further? There was also a really great article in the Boston Globe that was, think about teen speak. One of the best strategies is to have discussions, that Socratic dialogue. What would happen if? What do you think if? So on and so forth. And so what do? We help protect them. We help protect ourselves. And there are things that are not surprising. So things like good nutrition. Restful sleep is really one of the top things that if you can protect your sleep, because when you're tired or sleep deprived, that frontal lobe that's trying to hold back the want and need to read or have good cognitive control or have what were you thinking moments is more compromised when it's tired, because the frontal lobe takes a lot more energy to hold something back than does just making any other response. So if you can place a emphasis in any one particular area, and there are a lot of data to support this, is that if we could protect sleep, which means turning off screens so that the brain can actually wind down, rest, restore, that might be one of the best strategies for protecting brain development as well as reducing risk from addiction or other mental health issues. And so finally, nutrition, stable and nurturing relationships, people matter. We can't have enough people who care about us and who matter to us. And loneliness is really an emerging field too, where we think about we might seem more connected, but we are social creatures. Humans are social creatures. So we have to maintain and practice those connections with other humans. Stimulation and enrichment, the more things you can bring to your brain. People used to think that the brain just stopped. And one of the things we learned was that we have something called neurogenesis. The memory center of our brain can actually form new memories. And I think that is really promising. The brain can also recover. So if someone has an addiction or had a mental health illness, we know that there can be recovery. And even if the frontal lobe or the amygdala or other parts of the brain were impacted, they can recover with time. With abstinence, the frontal lobe can get back online to better decision making. We know that we can have a restoration of brain chemistry. We know that with mental illness, with treatment and with correct management, we also can help to facilitate restoration of the brain. Safe and supportive environments, of course. Physical activity and exercise. Everybody, if they can get out and move their bodies, because we are just sitting a lot of the times on screens. But again, that is one of the pieces of a digital technology age, is that we have less time. So let's use our resources, our cognitive resources, to figure out ways to get more physical exercise, quality sleep, positive social and environmental learning, healthcare and early intervention services, and finally, cultural and community connections. It's important for us as human creatures to have social connection to one another. And really, one of my favorite parts is, how do we cultivate joy? If we could find ways to cultivate joy, this character, if we can help find ways, because it's not about suppressing the negative emotions, it's about building up the good ones to have an impact on how brain functions. In those things, if we think about the takeaways, adolescent brain development is a time of rapid development over a very short span of time. If we are lucky and we live into 80s, 90s, maybe 100, what happens in our brain for the first 20 years is going to set that time course. Obviously, in the first 10 years, when all of our senses are coming online and integrating, that's really important too, and that needs to be protected. That's why early childhood adversity has such a big impact on brain development. But the things that happen in the second decade of life, when the frontal lobe is coming online, this is a very important time. It's almost like we need to develop some sort of a helmet, proverbial and actual, to be able to protect this process. But the most significant leaps in cognitive abilities also occur in that same second decade of life. So again, it's hard to imagine, oh, if I protect my brain now when I'm 10 to 20, that's going to put an extra 10 years onto the life of my memory. Because we're really good as a society in fixing a knee or fixing some other part of the body. Where we haven't been as successful is how do we keep the brain young? And really that's going to be the next decade of research, is how do we promote good brain health? And of course, this is a passage of time when emotions are strong and can be really hard to manage. When I give these presentations in person, I say, for better or for worse, we've all been adolescents, and everybody takes a moment. They're like, oh, I remember some of those really hard times. And this is a really important time for social emotional learning. So it's not always a better or worse, or a do this better or different. And certainly there is no shame in feeling emotions. Part of the process is to actually feel them, name them, and add them into some of the processing in the brain development. And so hopefully this is a very big body of literature that's been, interpreted. The neuroscience has been interpreted for you to understand that there are neurobiological risks. This is not a moral failure of someone who has a risk or becomes addicted or develops depression. This is that the teen brain is particularly more vulnerable to things like addiction or mental health issues because of the biological process itself. And so knowing family history, if there is a family history of addiction, there's a lot of mental health stigma, but sharing that information can be the difference about whether or not you can help protect offspring. Younger first use, the longer you could wait to start using. If you've already started using during adolescence, the quicker you can stop and give your brain time to do its developmental process. All of these things are what we've learned from the literature. These are not personal opinions. These are best practices, and these are ideas. Not everybody is perfect all the time. We live in a very complicated, complex world, and things obviously like sleep, exercise, and nutrition are important. And the last point here is that psychoeducation, and that is, one of my most important passions of my career is to be able to provide psychoeducation. So not only a science approach, but it's a biopsychosocial approach. How do we consider all the variables and really have this idea that understanding brain development, environment, but also diversity, and how do we learn coping strategies are all the things that we want to take together and educate ourselves with. So the goal is to really take all of this information, this psychoeducation, to be able to take this biopsychosocial approach where we work with the neuroscience field to be able to understand what the data look like, being collected in people with the issues and the concerns that they're having, and tie this together with things like psychology and understanding mental health and wellness, but also the social aspects. So how do we integrate with others? With the goal being that if we can understand brain development, because we all go through this process, we can use this as a tool. And we can integrate that with how we interact with the environment, how we think about diversity, and ultimately the goal in delivering this information today is to help to promote and to learn about healthy coping strategies. So thank you very much for your attention.